Okay, here we go with our last lesson for this module, Module 8. And so far, what we've talked about how cells replicate. We've talked about cellular reproduction. We talked about mitosis, and that's the main form of cell reproduction that happens all the time in your body for normal growth, normal repair, and so forth. But we also talked about meiosis, and that's about sexual reproduction, and that's actually about creating offspring. And that's about creating the sperm and the egg, the spermatogenesis and the oogenesis. And we looked at the mechanics, and we compared and contrast the differences between mitosis and meiosis. Now we're going to talk about what if something goes wrong. What if something goes wrong within the chromosomes in these processes of mitosis, and more specifically actually meiosis? What happens if something goes wrong in actually creating those gametes, those sex cells, the sperm and the egg? So what happens if, for some reason, there are changes in chromosome number? For example, one example would be nondisjunction. That occurs when both members of a homologous pair go to the same daughter cell in meiosis 1, or when a sister chromatid fails to separate in meiosis 2. Well, let's think about what that really means. So I'm going to take the first example, and when a homologous pair, if I draw this out real quick, homologous pair from meiosis 1, if they don't separate. So where's my pen here? Oh, there it is. Oops, here we go. So here's my initial pair of homologous chromosomes. And we're going to say this is meiosis 1. We're only looking at one pair of chromosomes. So here's our original from the parent. And what's that going to do? That's going to give us a separation through normal meiosis 1. And then what should happen through meiosis 2, as you all well know by this point, okay, should look something like this. And we have our haploid, 4 haploid gametes. That's what when it goes right. But what if, once again, in meiosis 1, both of these initial ones up here go to just one direction? Well, what then would happen? Well, that would actually be pretty devastating. So let me grab my eraser over here. I'm going to erase this and erase these guys, because if you think about it, what it would look like is in meiosis 1, once again, they both go to one side of these intermediate daughter cells. And then the end gametes down here, you'd have double the chromosomes on this side and 0 chromosomes on this side. That's if the problem happens in meiosis 1, earlier up in this chain of reaction. So obviously that would be more devastating. What if it happened a little bit lower down the chain? So let me bring up my eraser again. And let's say the problem wasn't with meiosis 1. Meiosis 1 worked just fine. But let's say the problem was with meiosis 2, and that we didn't get good separation here. Well, in this case, we'd have two, say these are eggs, 2 normal eggs. Then we'd have one egg with double the chromosomes and one egg with no chromosomes for these specific ones that we're looking at here. So once again, we can see that this would be potentially rather devastating. Remember, these chromosomes are what give you your genetic traits. They also, by definition, they hold the DNA, which are going to code for proteins. So if you are missing a piece of DNA or a chromosome that's going to code a protein, well, that can obviously be a big problem. Or let's say you have extra chromosomes to code a protein, especially during development. That can be a big problem. Specifically, once again, let me erase this here. We have, for example, with this non-disjunction, monosomy, which means we are missing a copy of one chromosome. An example of that would be Turner syndrome, where you only have one X chromosome, you don't have a second X, or you don't have a Y, so it's a problem with the sex chromosomes. Or trisomy, we have 3 copies of a chromosome. The very common example there is Down syndrome, where you have 3 copies of chromosome 21. So there's actually a rather famous young man with Down syndrome, because he actually became, even though he had Down syndrome, a rather popular actor for a while. His name's Burke. And once again, he has very common features with someone with Down syndrome. He's more of a rounded face. He had these folds under his eyelids. He's got people with Down syndrome have a swollen tongue, so it's a bit harder to understand them. But then also, because of this extra chromosome 21, we have extra expression of specifically what they believe is the main culprit. This was known as a GART gene. The extra expression of this gene throughout development leads to what? Mental retardation. What happens specifically? Let's go back to changes or problems with sex chromosome number. So once again, Turner syndrome, I already mentioned. Once again, we don't have an XY or an XX, we only have an X. Once again, they're only giving off a female expression, but not fully like they're supposed to. So you have short stature, broad shoulders with folds on the neck, underdeveloped sex organs, no breasts. You have Klinefelter syndrome. So that's where you have XXY. So wait a minute, XX is usually female, XY is usually male. Well, what do we have here? Well, underdeveloped sex organs, breast development, large hands, long arms, and legs. So literally both some male and female elements here. What about polyfemale? We have triple X or quadruple X. Well, with triple X, once again, you're getting overexpression of that extra X chromosome here. So it tends to be tall and thin, but not usually mentally retarded. However, if you have that even a second extra one with XXXX, they tend to be severely retarded. And once again, why are we having this mental retardation? It's because we have extra expression of the genes that are on that chromosome during development, anywhere from all the way from fetal development through adolescence and so forth. When your nervous system is developing, especially in the fetal development, and the extra expression of those proteins are actually going to hinder normal neural development. Lastly, we have Jacob syndrome. So we have XYY. So once again, same concept here, where we have extra expression of those genes on the Y chromosome. They tend to be tall, persistent acne, speech and reading problems. What if, instead of not a problem with meiosis, what if there's a problem somewhere along the line, instead of a whole chromosome going one side or other, what if there's a problem within a chromosome? Let's say there's just a deletion, which means a piece of the chromosome is broken off, if you will. That would be an example of what's known as Williams syndrome. That means, once again, we're missing the genes that are meant to be expressed on that chromosome. Or what if we move those genes that are on a particular chromosome from one chromosome to another chromosome that's non-homologous? Just they flip, if you will. They potentially just switch places. Or we have duplications of those genes within one chromosome, which all of a sudden that gene would be longer, but you have more, those particular traits are going to be expressed more. Or you have inversions. What if we literally flip that chromosome 180 degrees? So let's actually look at some animations on how this happens. Actually, before I get there, I forgot I have this slide. So this is just picture straight out of your book once again. So over here on the left, an example of deletion, once again, how the chromosome is supposed to look like. And what if this A simply gets deleted and broken off? Or duplication, once again, D and E right here are duplicated, which means we're going to have extra expression over here. Or what if we have an inversion? What if we flip C and D? Or translocation, where Q and R and G and H trade places. And once again, in this case, these chromosomes, notice they're non-homologous chromosomes, and yet they're flipping at their ends right now. So the genes are literally located on the wrong chromosome. You can imagine how devastating that would be.
An inversion is a reversal in the order of a segment of a chromosome. Inversions do not alter gene expression unless the breakpoints on the ends of the inversion occur within a gene. When a segment of a chromosome is inverted, it can pair in meiosis only by forming an internal loop. Although crossing over occurs in the 4-strand stage during meiosis, only 2 strands are shown for clarity. Crossing over that occurs within the inverted segment during meiosis can result in some genes being lost on one chromosome, while others are duplicated on another chromosome.
So once again, I imagine that you could think how devastating that would be if all of a sudden this were happening in meiosis, as it's stated, where one gamete, one egg, let's say, is going to have this extra long chromosome with extra expression of these particular genes, while the other one is actually literally going to be missing the genes that need to be expressed on that chromosome. And here's a summary of kind of all of those various changes in chromosome structure.
A chromosomal mutation is a substantial change in the structure of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. In the chromosome, shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in two places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve two chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
All right. Well, we're coming to the home stretch, only 2 weeks after this one left in the quarter, and I want you to realize how much we've gone over so far. I want you to once again always go back to our pyramid and realize that we've covered the biochemistry, we've covered the genetics in our studying and discussions about biological function. And we started our conversation on molecular biology. We've now discussed how DNA can replicate. In the next module, we're going to talk about the rest of what's known as the central dogma of molecular biology, how DNA actually encodes proteins. And then the last module, after we talk about this element right here, the gene expression, another way of saying it, we're expressing these genes to make the proteins. We're going to talk about, go back to the concepts we talked here about what happens with mutations in normal, say, mitosis, where we have uncontrolled replication over and over again, and that's cancer. So that's it for this week. I hope you enjoyed it.